Well, good afternoon, hobbits. Welcome. I have a few things to say to you before we begin. You are currently on a journey, a quest, if you will, to biochemistry. And this can be a difficult and challenging and dangerous quest. But take heart. You have help. You have people to guide you on this journey to biochemistry. But I know that you may be nervous because something very dangerous and great is looming before you. And this may have you trembling and afraid. Now, I understand this, but you can do it. Fear not. Now, at some point during this mighty test that you're about to face, you may question whether I've been true and whether I have been faithful to you. You may think that I have turned to the other side, but do not take me for some conjurer of cheap tricks. I'm not trying to trick you. I've been trying to help you. So, remember, you will face this test, this great battle, tomorrow afternoon. You should know where you should take up your battle position. This has been communicated to you. And remember, there are details here. This is not for you to decide, these details. All is for you to decide is what to do with the time that is given you. Now remember, you definitely must bring your student card to this test. Because if you do not bring your student card, then you shall not pass. Furthermore, make sure you have your weapons, proper weapons for the task at hand. Anything, these magical devices that I see people using, I don't know what they are, but you must not have them with you. You're not allowed to use these things. Keep them secret, keep them safe, put them away. And last of all, remember, do not despair. Despair is for those who know the end beyond all doubt, but that is not you. What you should do now is take courage for the storm that is at hand, and that is what will protect you and save you. That and listening to the advice of your elders that we've already given you. So take courage, and if things go well, you will have a happy ending to this dangerous quest. Okay. I'm not going to do the whole lecture like this. So give me a moment and I'm going to take off the beard. Well, thank you. Thank you. It's a small crowd, small but hearty crowd to go on this quest. All right. I got to go behind here to take my mask off. Yeah, surprise, it's me. Okay, so here we go. Back is me. Yeah, so just remember, go to the correct room, bring your student card, and everything's going to be fine. So, but today, because I need to use all the lecture time I have, because Dr. Heinemann is expected to take over soon, I want to continue on with topic 14. So, and this is where we're dealing with nucleic acid structure, and this comic really, you know, it mentions DNA, so I thought it was okay to put up there and somewhat relevant. So this is now stuff that will not be on the midterm, but is now on the final examination, where we're moving into information flow, transcription, translation, and that sort of thing. Okay. So in topic 14, we're going to review the chemical structure of nucleic acids, B, DNA, and RNA structure, what the difference is between DNA and RNA. We'll talk about stability of the double helix a bit, and we'll talk about DNA packaging. And so just to reorient you, right, we've talked about amino acids and we've talked about monosaccharides and lipids, but now we're moving into the fourth class of biomolecule, which is, of course, nucleotides and nucleic acids. So that's where we're headed now. And a lot of this stuff I talk about today is probably going to be reviewed because you've seen this before, but you may pick up some new things as we go along. So just try to focus and stay attentive. So what are DNA and RNA doing in your cells? Well, DNA, you know, is the information that you need to make the organism. It's basically an information repository. That's what DNA is good for. That's all it does. It's a source of information you need to build an organism. But for that to be useful, you need to express it or transcribe it. And that is mediated through RNA. So for a gene, you will transcribe it into messenger RNA, which gives the instructions to make the protein. And of course, that is mediated by other types of RNA, transfer RNAs, which bring the amino acids to the ribosome. The ribosome itself is made up of ribosomal RNA. And then there are other RNAs, smaller RNAs involved in this process, involved in splicing to, you know, shuffle the exons as needed, and in regulation, microRNAs and small interfering RNAs. Dr. Heinemann is going to talk a lot about transcription and these processes, but I just wanted to, you know, reinforce that RNA has quite a few functions related to gene expression. And so there's different types of RNA doing different jobs in the cell, whereas DNA only has one job, just be the information source. Okay, so that's why obviously it's important to understand the structures and behavior of DNA and RNA. So we'll start with the basics. You know that nucleotides are based on a sugar, ribose. So this is a pentose. Remember, a little review here. A pentose. This sugar, is it in the alpha or beta configuration? Anybody? I think I heard beta. That's right. So it's beta because the anomeric carbon has an OH that's on the same side as the highest numbered carbon. So this then forms the basis of ribonucleotides. And instead of adding a sugar onto the anomeric carbon, we add a nitrogenous base. And this is just one example of such a thing. This is called cytosine. Cytosine refers to the base itself that I've added onto the sugar. But if you want to refer to the whole molecule, that's cytidine. So there is a bit of different nomenclature. The words slightly change when you're going from the base to the nucleoside. And that's what this is, a nucleoside. A nucleoside is when you have a sugar and a base attached to it. So the whole thing is called cytidine, but if you just refer to the base, it's cytosine. And this is an N-glycosidic bond. Okay, where I talked about O-glycosidic bonds joining sugars together. This is an N-glycosidic bond. It's the same basic idea. Once you form this bond, the sugar is no longer able to switch back and forth between the alpha and beta configurations. And so all the sugars that you're going to see in DNA and RNA are in the beta configuration. And they don't flip back and forth. And now we need a way to refer to specific atoms in the molecule. And the convention is to number the base first and just call those numbers 1, 2, 3, 4, 5, 6. Don't worry about the numbering of the base. You don't have to memorize that or anything. I just point this out because then we want to number the sugar, and we number it the same way as I've already described for carbohydrates. But we put the prime symbol next to it. So it's 1' and 2' carbons and the 5' carbon. And that's to distinguish those atoms from the atoms in the base. In case you ever wondered why we say, you know, 3' and 5' and the prime is for this reason. So you can distinguish these atoms in the sugar from the atoms in the base. Okay, so that's a nucleoside and a ribonucleoside. When you add a phosphate to it, it's called a nucleotide. So the S turns to a T, and you just have to know that. So when you see nucleotide, that implies that there is at least one phosphate on it. And so if we wanted to name this, we'd call it cytidine monophosphate. It's cytidine with one phosphate attached to it. This could be called a nucleotide or a ribonucleotide, or you would call it a nucleoside monophosphate.
not a nucleotide monophosphate, a nucleoside monophosphate, because it's a nucleoside to which you've added one phosphate. Okay, so that's the correct way to refer to these molecules. And as you see, sometimes people will just full out say it's a ribonucleotide, but sometimes people get lazy and it's called a nucleotide, and you just have to know from context whether they mean a ribonucleotide or deoxyribonucleotide. And you can add one phosphate, you can add two phosphates to make the diphosphate form, you can add three phosphates. We've seen this idea before with the adenosine triphosphate, right? And the adenosine diphosphate, adenosine monophosphate, okay? And so those are all nucleotides. Those are ribonucleotides. These are the basis for RNA. And then for DNA, it's very similar. It's the same basic idea, except that the sugar has lost an oxygen on the 2' carbon. So it's 2' deoxyribose. And so they're deoxynucleotides. Or sometimes, again, people will just forget that and just say nucleotides when they mean deoxy. And again, a nucleoside triphosphate, often, if it's important, you would say deoxy, but sometimes you're lazy and you just say a nucleotide. So sometimes it's just based on the context to know whether it's ribo or deoxyribo. Okay, but the same type of structures form. Okay, so that's the individual building blocks. Now there's different types of nitrogenous bases you can put on there. The first type is called purines. This is the molecule purine shown on the bottom. And you can see it's got two rings joined together and a bunch of nitrogens in it. And adenine and guanine then are two variants on this purine structure. They've just got other groups added to them. You don't have to, again, don't remember the numbering, it doesn't matter. Although I will just point out that it's nitrogen 9 that is used to attach the base to the sugar. A couple things to note, though, about these structures. You can see they have hydrogen bonding donors and acceptors, various hydrogen bonding donors and acceptors. You know that's going to come into play when we talk about base pairing. But you also see that the structures are fairly flat in their ring structure. So it's kind of a flat molecule, the purine, and it has quite a few pi electrons in those double bonds, right? And so that's going to come into play later when we talk about DNA structure. But sort of a main point I want to make now is the surfaces, like the flat surfaces of these purines, are fairly hydrophobic. They're relatively hydrophobic. So these molecules have polar groups on them, and they're good hydrogen bonding donors and acceptors, depending on which group you've got. But when you look down on the flat surface, it's relatively hydrophobic. And that also comes into play when we're going to talk about DNA structure. Okay, then the other type of base that you see in DNA and RNA is pyrimidines, based on the molecule pyrimidine, which is shown at the bottom. And there's three main variants of this. As you know, adenine and guanine are found in DNA and RNA. Cytosine is found in DNA and RNA, but thymine is mainly found in DNA, and uracil is found in RNA, but not DNA. And you can see that thymine and uracil are very, very similar to each other. They're just the uracil is just missing this methyl group that is found on thymine. Otherwise, they're identical. And so by the same token, these also have polar groups that are good for hydrogen bonding. They're also, though, fairly hydrophobic when you look at them on their flat surfaces. So I've shown you the bases. Just to lay your mind at ease, you don't have to memorize the structures of the bases, okay? You should be able to tell the difference between a purine and a pyrimidine. That should be relatively straightforward because one has two rings joined together and is larger than the other. And you don't have to memorize the numbering, you don't have to memorize the structures. You do have to know the names and the symbols. You should know that C stands for cytosine, but hopefully that's fairly straightforward. Okay, so those then are the different groups that are attached to the sugar to make nucleosides and nucleotides. Okay, so those are the basic building blocks of polynucleotides. And then you can join those together in longer and longer strings. What we're seeing here is just a 4-base single-stranded nucleotide. And you can see how the sugars are joined together through this phosphodiester bond that joins the 3' carbon of one sugar to the 5' carbon of the next. And there's a phosphate group in the middle making that covalent linkage. And that's a phosphodiester bond. You can see that this is a strand of DNA because it's lacking the OH on the 2' carbon, just has a hydrogen there. And so you'll see then when you have this repeating nucleotide structure, there's a sugar phosphate backbone. That's what we call it. It's just repeating alternating sugars, phosphate, sugar, phosphate, sugar, phosphate. So it's the sugar phosphate backbone. This is analogous to the polypeptide backbone, right? Where you've got the peptide bonds and the repeating units of atoms. Same way we have repeating atoms in the backbone, and every nucleotide is the same in that way, and they differ in the base that's on them, which is sort of analogous to the side chains of the polypeptide, right? Where you've got the amino acid residues with the side chain sticking out, okay? And so, and like proteins, there is a difference between the two ends, right? The proteins has an amino terminus and a carboxy terminus, an N and a C terminus. Here we have a 5' end and a 3' end, and those are identified then by where it stops being joined to something else. So here the 3' carbon is not joined to another sugar, so that's the 3' end, and by the same token, at this end, we've got 5' carbon is not joined to another sugar. And sometimes students seem to think that the 5' end always has to have a phosphate on it, and that's maybe how you identify the 5' end, but that's not true. If I were to take the phosphate off of here, that would still be the 5' end, right? Because that's where the sugar phosphate backbone ends at the 5' carbon. And even if I added a phosphate down here, which you could, that's still the 3' end. So don't go looking for the phosphate to tell the 5' and 3' end. Look at the structure of the sugar, and you should know all about sugar structure now to identify which is the 5' carbon, which is the 3' carbon, okay? And we'll talk about how these are synthesized in topic 15, but for now, just take it for granted that we can make polynucleotides. And you can make RNA or DNA depending on which you're using, ribonucleotides or deoxyribonucleotides. Okay, and the last thing I want to mention this is how do you represent the sequence? You can see we've got different bases joined here in a specific order, and we want to be able to specify that order. So if we were to look at the sequence of this molecule, we would say that it is AGTC, based on the order starting from the 5' end of the bases. So that's the convention. When you see a nucleotide sequence, you can assume that it's starting with the 5' end of the molecule. Okay, just the way that protein sequences are written N to C. Nucleotide sequences are written 5' to 3', unless it's otherwise indicated, right? Unless someone tells you, well, the 3' end is here, then you know. But if it's not specified, always assume that the first one is at the 5' end, okay? Okay, so now double helix, you've heard of it. This is one of those things that's in the public consciousness, right? People in the street would know what you're talking about when you talk about double helix. They're like, oh yeah, DNA. So this was proposed in 1953 by these two people, and they ended up winning a Nobel Prize for this insight that they had because before this, people were very curious about the structure of DNA, what it looked like, and people really didn't know. But these guys got some inspiration, and they figured it out. But they couldn't have done it on their own. And that's partly because they weren't such great experimentalists. They did not tend to go into the lab and do a bunch of experiments to try to figure things out. They were the type of guys who would prefer to sit in an office and drink coffee and talk and just chat and theorize and do that sort of thing. So they weren't such great experimentalists. So they needed evidence that other people who did go in the lab and actually do stuff, the information that they provided. So the three main pieces of evidence that they put together to propose a double helix structure. And the first is Chargaff's rules. And these said that when you analyze the DNA composition of an organism, it's true, it tends to be true that the amount of adenine and thymine matches each other, it's the same, and the amount of guanine and cytosine matches each other. And you can see this is some data that's from this book that was published, and it shows that in a given organism, you know, the A and the T the percent A and T is the same or about the same. Now they're not exactly the same, but give the guy a break. It was 1950s, 1940s, and 50s, so you know the technology was not as good as we have today. So otherwise, it's pretty good data. And if you go from one organism to another, the percents differ from organism to organism, but within an organism, if you go to different individuals in that organism, it'll always be the same. right? And it won't change as the organism ages. It doesn't change whether the organism's male or female. It doesn't change even if you go to different tissues within the same type of organism, it's constant, okay? And obviously now we know what this means, but at the time it was interesting, but they couldn't quite grasp the meaning or the implication of this, all right? And so I did want, I like to show pictures of people. So this is Erwin Chargaff, who did the work and came up with Chargaff's rules. I don't know if he always wore a suit like this, but I think he looks pretty good in this photo. So that's the first thing that everybody knew, but nobody knew what it meant. So then the other thing was this. And this is famously the X-ray diffraction pattern that Rosalind Franklin acquired of crystalline DNA. And this, if you understand what these dark images and these shadows, what they mean, you can get information from this. And so it turns out that Watson and Crick were thinking about DNA structure, and then they were invited down to London by this fellow, Maurice Wilkins, who shared the prize with them in 1962. And he was a crystallographer working in London, England. And he was working on crystallography of DNA, but in fact, he didn't acquire this image. This was acquired by Rosalind Franklin. And so but what he did, he knew that she had acquired that image, and he showed Watson and Crick the image without her knowledge or consent even. I mean, all these people knew each other, but she happened to be not in the lab at the time when they visited. But he said, "Hey, did you see the latest great image that Rosalind Franklin got?" And immediately Watson looked at this and was like, "Ooh, that just tells me some information." And he couldn't wait to get back to his office to start working on that. So Maurice Wilkins won the Nobel Prize, shared it with Watson and Crick in 1962, even though he didn't acquire the data, but he was working on it. And in fact, Franklin is the person who got the data. And so you can see her dates here, right? So unfortunately for everyone, she passed away of cancer as a fairly young woman. And so she did not live to see the Nobel Prize being given out. And as I said before, they don't give Nobel Prizes posthumously. They only give it to people who are still alive. So it stands to reason that had she been still alive, she would have shared in the prize instead of Wilkins. But as a nod to the role that X-ray crystallography played in determining the structure, Wilkins did share in the prize. Although, obviously, I think it should have been Franklin who was honored in such a way. And so if you're going to read a book about the DNA double helix and the role of Franklin, I would really suggest this one, Rosalind Franklin DNA, by a friend of hers, Anne Sayre. And I think Anne Sayre wrote this, from what I understand, sort of as a counterpoint to James Watson's book, The Double Helix, which is his memoir of what happened and what it was like to figure out the DNA double helix. A lot of people objected to his characterization of Franklin in his book. And so Anne Sayre, I think, wrote this as sort of a way to set the record straight. So if you're going to read just one book, I would recommend this one. It might be interesting to read both of them and compare and see what you think. Anyhow, so that's the X-ray diffraction pattern. But I have to say, though, that Franklin had acquired these data and these images, and she was trying to figure out what it meant, but she did not actually have the brainwave to actually. Now, she would have, I'm sure, sooner or later. I have given enough time, but just Watson and Crick beat her to it. Maybe because they were not as hardworking in the lab, they were sitting around drinking their coffee and thinking. Anyhow, so Watson and Crick are the ones who actually proposed the structure, and Franklin in her notes, it looks like she was getting close or getting there, but she didn't quite bring it all the way to home. And then the last thing, though, that Watson and Crick didn't understand or didn't know that they needed someone else to tell them about was the correct tautomeric forms of the bases. And I wouldn't spend too much time worrying about this. I'm just saying it for more historical interest. But there are two different forms. If you talk to a chemist, there's two different structures you can draw for these bases. And some examples are here. And they didn't know which one they should be messing around with, because they had the idea they were going to talk to the people in the machine shop and get them to make models, like physical models of the bases, so they could play around with them, but they wanted to make sure they made them in the right chemical format. And so they had to ask their office mate, Jerry Donahue, who, and here's his picture, And he was a chemist, and he could tell them that, in fact, they should be looking at the keto form and trying to fit that together, because that would be the most stable form under the conditions they were interested in. Okay, so that's what they did. They got the machine shop to make a bunch of little models. So molecular models are important, and then they messed around until they realized the hydrogen bonding pattern and how that would work, and then they figured out the double helix structure. So, but they needed those information from other people to do that. And when I saw this picture of Donahue, he seems to know how to have a good time. I don't know what he's holding there and there, but it looks fun. And I thought, he looks a lot like Dr. Dempsey, who's our education coordinator. So if you've ever communicated with Dr. Dempsey, I don't know, maybe you see the resemblance there. I looked at it, I was like, wow, it's Brian. Anyhow, so Watson and Crick came up with this structure. And so the idea is you've got the sugar phosphate backbone, which is in gray here, and two strands come together, and the bases will base pair with each other, and that's seen on the upcoming slide. But the point is that the two strands associate with each other, and instead of staying in this sort of ladder structure, they'll twist around each other, and that brings the bases close to each other so they can actually touch, because in this ladder structure, they'd be too far apart to be touching. So you need to squish them together a bit, and that's what the helix accomplishes. And so a more accurate sort of 3D representation is shown on the right here, where you can see the sugar phosphates in white, and the colors represent the different bases in the middle. Okay, so just note though that the two strands come together in what we call an anti-parallel fashion, right? The 5' end of one is next to the 3' end of the other. So if you're looking at directionality, you go 5' to 3' down on the left strand, but up on the right strand. Okay, so it's and pretty much in DNA, it's pretty much always going to be this way, anti-parallel. That's the way the strands prefer to come together energetically. And they make the base pairs, and those are in the middle. The sugar phosphates are on the outside. And it is then wrapping, the strands are wrapped around each other in a particular fashion, what we call a right-handed double helix. And so this diagram here on the right tries to show you the difference between the right and the left-handed helices. You can see the twists are happening in opposite directions there. And I would expect a biochemistry student to know the difference and be able to look at this and say, oh, I can see that's a right-handed double helix. Well, how can you do that? There's different ways to try to remember which is which. One that I am partial to is if you imagine trying to grab on to the helix with your hand, and you'll see your thumb then, now my thumb is pointing up towards the ceiling. So if you follow along the strand, let's just look at the ribbon on the right, these red things. If you follow along the ribbon in the direction your thumb is pointing, you'll see your fingers are curving around in the direction of the right-handed helix. Can you see that? Whereas if you try to grab the left-handed helix, you see the curve, if you go up in the direction of your thumb, the curve is going the opposite way that your right hand is pointing. It doesn't matter if you go down or either. But if you try with the left hand, then that curves right. So there's different ways that you can remember. That's one way. Another way that I've heard that I kind of like is if you imagine you're at the top of it and you imagine it's a spiral staircase and you're walking down. If you're going down the right-handed spiral staircase, you'll be turning to the right. You can see that. And if you're going on the left-handed, you'll be turning to the left. So, and there's other ways too that people have, you can go online and see how people remember. But there is a difference, right? And it is important, alpha helices in proteins have a certain handedness. DNA helices have a certain handedness. It's good for those of us in life sciences to understand the difference, okay? Because this is practical, how these molecules exist. Question, yeah? No. So the prominent form is this bDNA form. There are different forms of DNA, aDNA, bDNA, there's zDNA, but what it most stably forms most of the time in your body is bDNA. And that's actually what was one of Franklin's great achievements, is that most people were crystallizing DNA under sort of dehydrated conditions, and Franklin got the correct conditions for bDNA, which other people were not able to do. But so no, left-handed, I'm showing it there as a contrast to what the bDNA is. And most of the time, if you're seeing DNA, you can assume that it's bDNA, which is right-handed. DNA can adopt left-handed structures, but we're not going to talk about that in this course. Okay, we're going to keep it simple. You know, I do talk about zDNA in the 3rd year course, so you can take 3382. All right. So now how is this stabilized? There are 2 major forces stabilizing the structure of a double helix to keep the strands together in the structure. Base stacking and base pairing. So base stacking just refers to the interactions that happen between the bases as they're stacking on top of each other. Remember I said the bases are pretty flat, and you can see that in the diagram there. Even the base pairs, they're flat, so they can stack on top of each other very easily, and they're relatively hydrophobic, I said. Remember there's those pi electrons, and they can form these induced dipole interactions, you know, so-called London forces, if you remember your chemistry, that will cause them to attract each other. And that turns out to be the major driving force for the stability of the double helix. A lot of people will assume that it's the base pairing, but in fact, that does not contribute as much stability as the base stacking does. And you can think about the hydrophobic effect, right? The hydrophobic effect, when proteins fold, they bury their hydrophobic regions in the middle. It's the same sort of thing here. The surfaces of the bases are hydrophobic, and you want to bury them in contact with each other. Okay. And those interactions then, any base pair can stack on any other base pair, right? So you don't need a specific sequence there. Whatever the sequence is, you're going to get base stacking interactions. And base pairing occurs across the strands. So the base on one strand forms hydrogen bonds with the base on the other strand. And it always is in this pattern, or practically always, A base pairs with T and G base pairs with C. So that explains Chargaff's rules, right? And every time you've got a G in a double helix, you also have a companion C, and same thing with T and A. So those are so-called monogamous. I like to call them monogamous. They don't, in DNA, they don't tend to cross base pair with other types of bases, and because that would destabilize the structure. And yeah, so you can make 3 hydrogen bonds between G and C, and you can make 2 between A and T. And you can see they're complementary. So this is the great insight that these are called Watson-Crick base pairs, right? Because Watson and Crick first figured out, oh yeah, this is how it works, and they have a constant length, right? And which makes them really handy to fit inside the regular double helix structure. Okay. So remember, base stacking is the strongest contributor to the stability here. Okay, so now I've given you some information, and I've been talking a lot about stuff you probably already had seen before, but just to make sure you're awake and just to see if you're absorbing what I'm saying, here's a question. With a small crowd like this, I don't know how many responses we're going to get. Maybe give you 10 more seconds, because this is one of those questions I think most people that look at it and they're intuitively drawn to a certain answer. All right. Okay, so numbers are fairly small, but we got about 20% picked A and about 80% picked B. Okay, so, and nobody picked anything else, so that's good. You recognize that A and T base pair together, but which is it? Which is it? AATTT or is it TTT-AAA? Well, here's where you got to remember the convention that I told you. Anytime you see a nucleic acid sequence, you have to assume that it goes 5' to 3' as shown. So that would apply both to the question and to each of these responses. Okay, so now you got to think and remember that the double helix is anti-parallel. So the 5' end of one is going to match up with the 3' end of the other. So if you put these things together, if you have one strand that's AATTT, the other strand will read AAATTT, also 5' to 3'. So 20% of you got this right. A is the correct answer here. And people are like, oh, that's a trick question. Well, no, it's not a trick question, because I told you, you always assume it's 5' to 3' whenever you see it, and it's anti-parallel in the double helix, okay? So this is a case where the sequence on one strand reading 5' to 3' is the same as the sequence on the other strand. And in biochemistry, we refer to this as a palindromic sequence. So a palindrome in Okay, now I'm not muted. But now the subtitles are showing. I don't like that. Okay. Let's continue. Oh yeah, well, why did I even bring this up? Well, one is just, yeah, to reinforce this idea of 5' and 3', the antiparallel stuff. But the other thing is palindromic sequence turned out to be interesting. They're the sites of a lot of protein binding. So there's proteins called restriction enzymes that will cut DNA, and Dr. Heinemann is going to talk about this later in the course. So I just want to set you up for that though. And restriction enzymes tend to cut sequences that are palindromic, like this one. Okay, and there's lots of different palindromic sequences you could come up with, right? But that's where proteins tend to bind if they're dimers especially, okay? But Dr. Heinemann will go further about DNA protein interactions and restriction enzymes, okay? So now let's just look at the features of the double helix. This is maybe a more accurate sort of model, although it's not accurate because it shows too much space in between the bases. Remember, they actually touch each other. But the points I want to make here are the dimensions of it. It's about 20 angstroms in diameter. And what's an angstrom? It's a unit of measurement. 20 angstroms is the same as 2 nanometers. So nanometer is 10 to the minus 9 meters. And so this is 20 of those, okay? Well, 20 angstroms anyway. The angstrom is 10 to the minus 10 meters. Okay, and so that's the diameter. And then you can see that what this is showing here is one complete turn of the helix. So if we start at this point where they sort of cross each other and you go one complete turn, that is about 10 base pairs. So that's a good number to know. 10 base pairs per turn of the double helix, and that spans about 34 angstroms, or 3.4 nanometers. So that means going from one base pair to the next is about 0.34 nanometers. And then you can see it shows the anti-parallel nature. You get the 5' and 3' ends opposite each other. And then it shows you these major and minor grooves. And that is sort of the groove is where the surface of the double helix, sort of goes in towards the central helix axis. It sticks out. It's sort of at its widest when you've got the sugar phosphates here, and that's what these red and yellow. The yellow is the phosphorus, and the reds are oxygen. So you got the sugar phosphate that forms sort of like a ridge going around, and then you've got the so-called valley or something, or a groove where there's space in between the ridges. And there are 2 grooves going the whole length of the helix, right? They wrap around the helix, so it's a single groove. You could stick your finger in there and just travel around and get from one end of the helix to the other. But one is called the major groove and one's the minor groove. And you can just see from the structure here, hopefully, that there is simply more space in between the backbones, the 2 backbones in the major groove than there is in the minor groove. You can see the minor groove is a little more cramped. Okay, not as much space there. And sometimes students, have trouble understanding why you have grooves of different sizes. And the best way I can explain that is to look down the axis. And that's what we see on the right here. The blue are sort of nucleotides that are in the distance, but the white one here is right on top that's highlighted. It's an AT base pair. And you can see that the sugar is here, and there's a phosphate and the sugar, and then the sugar is here, and the phosphate, and then the bases are here. But if you look at the way that the sugar phosphates are oriented, they're not completely symmetrical in terms of, evenly distributed. They're actually sort of more oriented down towards the bottom. So if we look at the bonds joining the sugar to the base, you can see that they're not completely opposite each other at 180 degrees. They're actually making an angle like this. And so on this side, there's less space in between the sugar phosphate backbones. That's the minor groove. And on this side, there's more space. That's the major groove. Okay. You can imagine if the structure was different and the bases were more evenly spread out, 180 degrees opposite each other, the grooves would have a similar size. But in fact, it's this way, and one side is more cramped. There's less space than the other side. And it turns out that the major groove is just the perfect size for an alpha helix of a protein to fit in. And that's one of those things that makes me go, very interesting, that a protein alpha helix, which is one of the major secondary structure elements of proteins, just fits perfectly in there, and the side chains can interact with the DNA, and the size is just perfect. An alpha helix cannot fit in the minor groove, but it does fit in the major groove. Okay. So, and what do I expect you to know? I do expect you to know these numbers here. It's not that many. It's the width of the thing, 10 base pairs per turn, and 34 angstroms or 3.4 nanometers per turn. Okay, just a few numbers. Okay, in the last little bit, I'm going to talk about nucleic acid stability. And when we talk about stability of nucleic acids, we refer to how prone they are to, the two strands are to coming apart. And so this diagram is showing transition, where on the left side, this is low temperature. On the left side, this is supposed to represent double helical DNA. But as you increase the temperature of that, the DNA will start to unwind. The two strands will start to come apart. And so we could call this denaturing of the DNA or separation of the strands, okay? But that happens when you heat, right? Because you're adding energy to the system, things get bouncing around more, and the two strands then start to come apart because the energy holding them together is overcome by this kinetic energy that's being put into the system. But you see this doesn't happen instantaneously. As you increase the temperature, we get a transition. And what we're following here is the absorbance at 260 nanometers. It turns out that single-stranded DNA absorbs light more at 260 nanometers than it does at than the double-stranded DNA does. So you can follow this. You can follow the absorbance at 260. You can see it rise as the DNA becomes more and more single-stranded. But it's not instantaneous. You have points in the middle here where, say 25% of it is double-stranded and 75% is, that would be up here, 75% is single-stranded, right? That's where we're 3/4 of the way to the top. But after you get fully denatured, the absorbance doesn't change as you continue to heat because you've done the transition. So you get this S-shaped curve, and the point at which it's half denatured or half melted, that's what we call the melting temperature. And so that's going to be in the middle of this curve. But keep in mind, if we say there's a melting temperature of whatever, it looks like at about 72 degrees or something, that doesn't mean that at 71, it's all perfectly double-stranded, and at 73, it's all perfectly denatured, right? This is a slow-ish transition. And the transition is reversible. So you can take single-stranded DNA that's complementary to each other and put them together and cool it down, and the DNA will re-anneal or renature to form double-stranded DNA. So this is freely reversible, this process. You don't usually need chaperones or anything like that. If you handle the conditions carefully and give it enough time to find the proper binding partners, the base pairing partners, you don't need any other molecules. DNA molecules themselves will properly renature if treated right. Okay, so that's freely reversible. I just want to get that across. And this is important, when we're talking about pulling strands apart when we do transcription or replication. But it's also important to know when we're thinking about biotechnology, because we've got recombinant DNA techniques that rely on our ability to make primers or make small oligonucleotides that anneal to other pieces of DNA. And say you're doing PCR, people know about PCR, right? You want to design primers that have to anneal. You have to know what temperature those are going to anneal at, so you can design your PCR properly. And so that's why thinking about this nucleic acid stability has real importance to a biochemist. And so we're going to just go over then a few factors affecting the melting temperature, because different pieces of DNA will have different melting temperatures, and melting temperature is affected by conditions too. So there's intrinsic factors, intrinsic to the DNA itself, and there's extrinsic factors or environmental factors that affect the melting temperature. And so the first three here are intrinsic factors. The longer your piece of DNA, the higher the melting temperature. And that stands to reason, right? The more base pairs you have, the stronger the double helix is going to be, the harder it is going to be to pull it apart. So the higher temperature you need to get to make it single-stranded. So the longer the DNA, the higher the melting temperature. Likewise, the proportion of GC base pairs matters a lot. And the more GC base pairs you have, the higher the melting temperature as a rule. And again, a lot of people would assume that this is because there's 3 hydrogen bonds in between G and C, and there's only 2 between A and T. But as I said before, the main factor that's driving stability of a double helix is the base stacking interaction. So it also turns out that GC base pairs have stronger base stacking interactions, especially dinucleotides that are both GC, GC. Those have stronger base pairing interactions than AT-rich regions. So it's true that the hydrogen bonds do play a role, but that's relatively small relative to the base stacking. But the fact is that the more GC content you have in your DNA, the higher the melting temperature, as a rule. Complementarity, this just refers to how exactly are the sequences complementary to each other. That's what we call it, right? We've got, you know, AAA, TTT on one strand, then it's AA, TTT on the other. That's complementary. They're complementary to each other. If you know the sequence of one, you also know the sequence of the other. So but if you put it the other way, as you start introducing mismatches, you're going to lower the melting temperature, right? Because then you're not getting as nice of an interaction. The base pairing is disrupted. The actual structure is going to be disrupted. The stacking can be disrupted. So the more complementary you are, the 2 strands, then the higher the melting temperature. Conversely, if you start introducing mismatches, then you're going to lower the melting temperature. So those all are inherent or intrinsic to the sequence of the DNA itself. And then we have these environmental factors. So as concentration of salt increases, that tends to stabilize the helix. And that's because the negative charges on the phosphates, I didn't point that out before, right? But phosphate has a negative charge. And you get these repeating negative charges in the backbone. And they're held together a little closer than they would normally like to be in a double helix. So when you add salt, that sort of neutralizes the charge. And you can put a positive charge in between those negative charges, and it stabilizes things. Okay, so as a general rule, as the concentration of salt increases, your melting temperature is going to increase as well. But the other 2 things here, as their concentration increases, it's bad for the DNA structure. So organic solvent, just like any organic solvent you can think of, even ethanol, will tend to destabilize the double helix. And that's because that makes the environment more hydrophobic. And as I said, the hydrophobic effect is important to keep the double helix together, just like proteins. As you make the environment more hydrophobic, the hydrophobic effect is lessened, and you tend to destabilize the helix. So the lower the organic solvent concentration, the better, the higher the melting temperature, so to speak. And then urea and formamide, I decided to put their structures on here. I don't think I put them on the posted slides, but these are just small molecules that are good at hydrogen bonding. And it turns out that they can get in between the strands and interfere with the hydrogen bonding of the nucleotides, of the bases. So as the concentration of urea, say, increases, that destabilizes the double helix as well. And conversely, as the concentration of urea goes down, you're going to increase the melting temperature or stabilize the double helix. And typically in an experiment, we wouldn't add urea unless we wanted to deliberately destabilize the double helix. Okay, so those are factors, and I would expect you to get the logic behind that. Hopefully it's fairly straightforward, so you can predict what the impact of different changes is going to have on the melting temperature. And then finally, as you go to extremes of pH, that will destabilize the helix and lower the melting temperature. But we're talking extremes here. You have to go pretty far from neutrality, like way up to, you know, 10 or 11, or, you know, down to 3 or 4, something like that. In the typical range, like 5 to 9 DNA, this has no effect. It doesn't really, the pH doesn't really affect. You have to get to the extremes in order to lower the TM. But what happens there, you can start changing the protonation state of the bases, and that will mess around with their hydrogen bonding properties. And so that's going to destabilize the double helix. Okay. So I would expect you to know those different impact of those different factors on melting temperature. Okay, so that takes us to the end of today. And so you got the midterm tomorrow. I wish everyone all the best on that. And after reading week, we will continue with topic 14. Okay, so I hope have a good exam, and then I hope you enjoy your break.